Okay, first we're going to be talking about autocrine signaling, which means that the cell is communicating with itself, and the signaling and target cell are the exact same, which can also occur in bacterial quorum sensing. Here, the signaling molecules are being released from the cell and are going right back into the receptor proteins of the same cell, which is going to activate itself. And here we're zooming in, which is going to show the signaling molecule growing, but it doesn't actually mean anything. It just is going to show the perfect fit between the signaling molecule and the protein receptor, which then activates the whole cell and triggers a cell response. An example of autocrine signaling is the growth of cancer cells, in which the base layer of cancer cells starts sending one chemical to the same cell in the body, and the cells can start growing rapidly and dividing. The cells have to rely on themselves for cell division since the body already wants to reduce the presence of these cells in the body.
Talking about the G protein coupled receptor. This occurs on the cell membrane and consists of a receptor, a G protein that has an alpha beta and a gamma subunit, and a GDP, along with a ligand and an active target protein. First, the ligand binds onto the extracellular part of the receptor protein, and at the same time, the G protein binds to the new signal receptor complex inside the cell. Once binding to the complex, the G protein GDP is exchanged for GTP, which has 3 phosphates, not 2, and it activates the overall complex. From there, the G protein breaks apart, leaving 2 subunits, but takes GTP and binds to and activates the target protein. Target protein is then able to make intracellular events, which leads to a cellular response.
An example of when this is used is during an adrenaline signaling pathway where heart rate is increasing and hydrophilic signaling molecules are involved. Along with that, G protein coupled receptors are turned off when the ligand dissociates from its receptor. Considering our example, the termination of the adrenaline signaling pathway would begin once the adrenaline dissociates from its receptor, undoing the rest of the process. The G protein coupled receptors contain hydrophilic signaling molecules that quickly travel to the cell and are short-lived and need a receptor on the membrane. On the other hand, hydrophobic signaling can diffuse across the membrane and are slow to travel, long-lived, and usually occur amongst intracellular receptor proteins because they are hydrophobic. Now we're going to be talking about antagonists of G protein coupled receptors. Antagonists are any ligand that binds to a receptor and inhibits or stops a response.
So in this case, antagonists of the same receptor protein binds to the receptor proteins in such a way that it blocks the ligand binding site but does not actually activate the receptor. This means no other ligand can bind to the receptor, and so the cell's response to the signaling molecule is suppressed.
And an example of an antagonist is caffeine, in which it is an antagonist of the adenosine receptors.